Hello, I am JD and you are listening to The Ongoing. You already know, more or less, how a vaccine works. Traditionally, you take the germ that causes a disease, make it safe, and put it into the body. So your immune system can learn to recognize it and fight it off later. But the traditional approach shares one problem. You have to get the actual germ. You have to grow it in large amounts, handle it, and carefully make it safe. And for the most dangerous disease in the world, growing barrels of the very thing you are trying to defeat is slow, costly, and risky. So people begin to wonder, what if you did not need the germ at all? What if instead of putting a piece of the germ into the body, you could just send in a set of instructions and let the body build that piece itself? That is exactly what an mRNA vaccine does. And this episode is the story of how mRNA vaccines were discovered and made. It is a great story, but a long one, so we are telling it in two parts. In this first part, we will cover where the idea came from and the first big problem scientists ran into trying to make it work. So let's begin. To understand this, you need to know a little about how your body already fights disease. Deep inside every cell is your DNA. Think of it as an enormous cookbook, holding the recipe for everything your body can build. From the proteins in your muscles, to the hormones that travel through your blood, to the defenses that fight off disease. But the cookbook never leaves the cell. So when the cell needs to build something, it copies just the one recipe it needs onto a short working copy, made fresh whenever it's needed. That copy is called RNA. The kind that matters for our story is called messenger RNA or mRNA. Name tells you exactly what it does. It is a messenger carrying a single recipe from the cookbook to the place where it actually gets built. Your body sends out these messengers, constantly telling your cells to build this, make that. One of the things your body builds this way is its defense against germs. When a germ invades, your body needs to build something specific to fight it. a protein called an antibody, shaped to match that exact germ. A different germ needs a different antibody built from a different recipe. So the cell pulls the matching recipe, sends out the messenger, and builds the antibody it needs, and remembers that recipe. So it's ready even faster the next time. Now, here is the thought that changed everything. If mRNA is just a messenger carrying a recipe, what if we could write our own? Think what that means for a vaccine. You write a message with the recipe for one small, harmless piece of a germ and send it in. The cells read it and build that piece. The immune system finds it and builds the antibody to match, exactly as if the real germs were there. So when the real germ arrives, the body is already armed. You never bring in the germ. You just send the recipe. And the message is far easier thing to make than a living virus. It was beautiful. It was simple. And it very nearly did not work at all. Because when scientists actually tried it, the body fought back. They wrote their mRNA message and put it into a living body, and the body attacked it violently. Remember, the immune system is always hunting for foreign invaders. And a loose piece of mRNA arriving from outside looked exactly like something a virus would bring in. So instead of reading the message, the body raised the alarm, set off a storm of inflammation, and destroyed the mRNA before a single instruction could be read. The message was being burned before anyone could open it. For years, this was a wall no one could get past. The funding dried up, and the most scientists gave up on the idea. But not everyone. Her name was Katalin Kariko, a biochemist born in Hungary who had come to the University of Pennsylvania. And she simply could not let go of the belief that this messenger could become a medicine. It is worth pausing on what that belief cost her. Grant after grant rejected. She was demoted, her salary cut. For years, she kept coming into the lab to work on an idea that the scientific world had, by almost every measure, decided was a waste of time. Most people, at some point, would have moved on to something safer. She did not. She teamed up with an immunologist down the hall named Drew Weissman. And together, they asked the question everyone else had given up. Why does the body attack this messenger so fiercely? And the answer, when they finally found it, turned out to have been hiding in plain sight the whole time. To understand what they found, you need one more detail about mRNA, about what the message is actually made of. An mRNA message is written in a chemical alphabet with just 4 letters. 4 different molecules called bases, strung together one after another like beads on a string. On their own, single letters don't mean much, but read 3 at a time in order. Each little 3-letter word spells out one specific building block called an amino acid. String enough of those amino acids together in the order the message spells out, and you get a protein, an actual physical thing your body can use. So the message itself is not the finished piece. It is the instructions for building it 3 letters at a time. Now here's the thing Carrico and Weissman uncovered. When your own cells write their mRNA, they do one small thing before sending it out. They make tiny chemical changes to some of the letters. Small tweaks. They don't change the message at all, but slightly change the letter themselves. I am going to call these little tweaks decorations, because that is the easiest way to picture them. Think of the letters getting small accent added on the top. The word underneath is the same, but it is dressed up. For a long time, no one thought these decorations mattered. They seemed like minor housekeeping. So when scientists wrote mRNA in the lab, they left them out and used the plain letters. Why copy something that seemed unimportant? But those decorations were the whole secret. They were a password. Your body decorates its own mRNA to sign it, to say, this message is mine, leave it alone. And the immune system had learned the rule. A decorated message is one of ours, but a plain, undecorated message is a stranger. That is exactly how a virus's mRNA looks. Attack it. The lab made messenger with its plain letters, had no password. So every time the body saw a stranger and tore it apart. And once they understood that, the fix was almost unbelievably simple. Decorate the lab mRNA the same way the body decorates its own. Give the message the password without changing a single word of what it said. So they did. And this time, the attack never came. The body read the signature, recognized one of its own, and let it pass. The cell picked up the message and quietly built exactly the piece the instructions called for. The first great problem was solved. They published it in 2005, and the world barely noticed. The idea still seemed too strange to matter. And so the breakthrough sat quietly on a shelf, waiting for its moment. But even a messenger the body would accept was not yet a vaccine, because a second problem was still waiting, every bit as hard as the first. The mRNA message is astonishingly fragile. Out in the open, it falls apart in minutes. And even if it survived, it could not get inside a cell on its own. It needed a vehicle, a shield to protect it and carry it safely into the cell. And the answer would come from an unexpected place. From a scientist who was not even thinking about vaccines at all. That is where we will pick up next time. That's it for today. Thanks for listening. I am JD and this is The Ongoing.